Hi, my name is Philip, and I will present a project called Systemic Inflammation Causes Neuroinflammation Following Human Traumatic Brain Injury. I have no relevant disclosures. The neuroinflammatory response has been shown to be a key driver of the secondary injury cascades following TBI. And animal models have shown that systemic inflammation causes a neuroinflammatory response. However, how systemic inflammation affects neuroinflammation following human TBI is not fully understood. There are three aims for this project. First, how does systemic inflammation affect neuroinflammation following TBI? Second, can the neuroinflammation be altered using anti-inflammatory treatment? And third, can blood compartments be used to monitor the neuroinflammatory response? This project is based on a previously conducted RCT. 20 patients with severe TBI were randomized to treatment with interleukin-1 receptor antagonist or with placebo. 42 cytokines were monitored using a Luminex assay. Concentrations were measured in brain extracellular fluid and pooled every 6 hours, as well as in arterial and jugular venous blood twice a day. CRP, white cell count, temperature, and confirmed clinical infection were used as systemic inflammatory markers. Linear mixed effect models, cross-correlations, and multiple factor analysis were used as statistical and machine learning tools. Here we see a heat map displaying the results of the mixed effect model. Each row corresponds to a multiple linear regression mixed effect model with a brain cytokine as the dependent variable. Along the y-axis, we can see all cytokines, and on the x-axis, we can see the predictors. Time, infection, CRP, arterial cytokine corresponding to the brain cytokine, interleukin-1 receptor antagonist treatment, temperature, and white cell count. Here is a similar heat map, but for the blood cytokines instead. We have changed the cytokine predictor to microdialysis cytokine, which is the brain cytokine. Now, if we dive into the heat map, we can notice that for the interleukin-1 receptor antagonist treatment, approximately half of the cytokines are blue and half are red. The blue fields denote a negative coefficient between the predictor and the cytokine, while the red fields denote a positive coefficient. For IL-1 receptor antagonist, we get a positive coefficient as expected. For IL-6, however, only the blood shows the expected decrease in concentration. And a similar picture is observed for IL-1 beta. For IL-10, we can see a decrease in both brain and blood. When looking at white cell count, we know that the brain cytokines generally have positive coefficients, while the blood cytokines have negative ones. Similarly, we see an opposing picture for infection, such as a ventilator-associated pneumonia, where the neuroinflammatory response is different from the systemic inflammatory response. Thus, it seems as if the systemic inflammatory response, proxied by these inflammatory markers, alters neuroinflammation. In order to investigate the temporal dimension of our data, we have done a cross-correlation analysis. In this figure, every point corresponds to the maximum cross-correlation between the brain and blood time series for every patient and cytokine. Each lag is 6 hours, and values here on the left denote a sequence of events moving from brain to blood, and values on the right denote a sequence of events moving from blood to brain. Our data is centered around lag 0, showing that we cannot observe any temporal trend for this 6-hour resolution, which probably is too low to detect cytokine or information fluxes. Multiple factor analysis can be seen as a grouped principal component analysis. Here, we can observe that arterial and jugular venous blood hold similar information content, thus monitoring the blood compartment closer to the brain does not provide any significantly different information in regards to cytokine levels. However, the information content from the brain extracellular fluid cytokines is distinctly different. So, we have shown that systemic inflammation alters neuroinflammation, indicating that ICU patients with infections potentially present with a neuroinflammatory response. Interleukin-1 receptor antagonist administration significantly alters different blood and brain cytokine levels, illustrating the complexity of the inflammatory cascades and suggesting a potent treatment effect. Jugular and arterial blood hold similar cytokine information content, but brain extracellular fluid is markedly different. Therefore, cerebral inflammatory monitoring provides independent information. Thank you.